Um, I guess we could start. I'll introduce you, and people will trickle in. I mean, they're not here to listen to me talk anyways, so I might as well start off um, with the introduction, and then you can take over. Um, Hello, everybody. I'm not Anna Del Prado. She is traveling, so I'm helming the seminar series alone. I'm Paul Meyer, and it is my pleasure to introduce the person that you actually came here to see, Dr. Shivon Robinson, who's our second speaker in the IBANGS seminar series this year. Dr. Robinson told me that she's actually from New York, from Poughkeepsie, but she got her BA in psychology and biology with highest honors in neuroscience at Williams College in 2011 in Massachusetts. She had already started her research there as an undergrad, looking at gestational exposure to allopregnanolone and its effects on affective behavior. Then she got her PhD at Penn with Irwin Luckey, where she did a lot of work looking at the neurobiological basis for the therapeutic effects of buprenorphine, and then graduated from there in 2016, and then went on to do a postdoc with Julie Blendy, looking at the effects of perinatal opioid exposure on a number of behavioral effects. And I don't want to give too much away. We'll hear about some of that today, I'm imagining. Dr. Robinson also did work with a mouse model of the opioid receptor polymorphism, which is really important and of particular interest to us IBANGS folks. I don't want to give any more away about that either. And then after her postdoc, Dr. Robinson went back to Williams College, and has been an assistant professor there since 2019, and tells me that she now has her lab up and running, which was challenging to do during a pandemic, but she's got a group of good people working for her, and is starting off her rising career. Speaking of rising career, just one award of note in 2020, she was given the Cell Presses, made the Cell Press list of 1,000 inspiring Black scientists in America in 2020. So I'm proud to, and the rest of us should all be proud to have known Dr. Robinson way back when, before her rising star rocketed through the stratosphere. Without further ado, Dr. Robinson, it's a pleasure having you here. Take it away.
Thank you so much, Paul. And thank you for having me here. I will be sure to remember you all once I've become a huge rock star scientist. So yeah, this has been a very circular process for me. I started at Williams as an undergrad doing research, looking at gestational exposure to drugs, and now I'm here again using a slightly different model, but definitely kind of ties into how my interests have developed over time, trying to understand how these early life experiences impact long-term development, but in this case, really trying to understand what's happening during that withdrawal process in an infant. So today I'm going to be talking to you about some work that I did while I was a postdoc in Julie Blendy's lab, trying to really characterize some of these neurobehavioral effects of early life opiate exposure, particularly in this OPRM1 polymorphism. So trying to understand how genetic variation plays a role in some of these outcomes. Oopsies. So as I'm sure you're all well aware, we are currently in the midst of an opioid crisis. So I like this figure because it really shows how dramatically opioid-related overdoses grew in a relatively short time span. And although there have been some structural changes in the terms of in how opioids are prescribed and dispensed, we still see a really alarming amount of people who are experiencing these overdoses. And in 2019, there was approximately 50,000 people who died from an opioid-related death. So it's clearly still an issue that's affecting us today. And with the onset of the COVID-19 pandemic and subsequent disruptions to the healthcare system, combined with other social and economic stressors, I'm sure we're going to continue to see this opioid epidemic rise. And in parallel with these trends, we've also seen a significant increase in the number of infants who are exposed to opioids in utero. So again, this is looking at a period between 2004 and 2014, but there was about a 5-fold increase in the amount of infants who were diagnosed with neonatal opioid withdrawal syndrome. It was also previously referred to as neonatal abstinence syndrome, so you might sometimes see NAS interchanged with NOWS. But this is a huge public health issue in addition to the overdose deaths, that we have this huge rise in infants who are exposed gestationally to opioids, are experiencing this withdrawal syndrome, and it's not really clear kind of what the long-term outcomes of that are. So to briefly kind of go over what this withdrawal syndrome entails, when human infants are exposed to opioids in utero, they do become physically dependent. And upon birth, this combination of withdrawal symptoms will manifest in that early postpartum period. And it really ranges from kind of this hyper irritability, incessant crying, to gastrointestinal distress. So you see vomiting, diarrhea, difficulty feeding in these infants, to sleep fragmentation and disturbances, to kind of tremors, hyperthermia, some more kind of autonomic symptoms. And these typically manifest about 24 to 72 hours after birth. And to date, there is no kind of standardized approach to how to treat NOWS. Each institution has their own way of dealing with it, but the most commonly prescribed pharmacologic intervention is opioid replacement therapy. So to continue treating the infant with either morphine or methadone until these withdrawal symptoms dissipate. And then if they're not responding to those initial first-line therapies, in some cases, they'll be given phenobarbital or clonidine. So what's interesting about this syndrome is that it is not consistent. So about 60 to 80% of infants who are exposed to opioids will require some type of pharmacologic treatment. Right, so there is some variation in its severity. And then infants who do have this more severe phenotype of NOWS require longer duration of treatment. So that's even more exposure to either opioids or these other classes of drugs that could be having an impact on their development. So one of the major questions in the field is trying to better understand the biological factors that are impacting the severity and the long-term outcomes. So there's only been a handful of studies in humans that have looked at kind of what are the long-term effects of that early life opioid exposure. But as you can imagine, there are many environmental, social, economic factors that could be influencing an individual's trajectory, and that it can be difficult or impossible to separate that from the drug exposure itself. So this is where animal models really become useful in trying to kind of identify the specific mechanisms that are at play, with the ultimate goal of trying to achieve more efficacious treatments. And so again, identifying either biomarkers or genetic propensities that can help in predicting the severity of the syndrome or potential long-term outcomes is really a big area of research interest. So within the Blendy lab, we had, there we go, had previously been interested in this gene variant that occurs in the mu opioid receptor. So this is the receptor that is the major site of action for both prescription and illicit opioids, including morphine, oxycodone, heroin, and fentanyl. It's fairly widely expressed throughout the central nervous system and the periphery, but we do see these distinct anatomical distributions in brain regions that we know regulate both pain and reward. On the right here is a schematic of the receptor itself. So it's a 7-transmembrane G protein coupled receptor. And there is a single nucleotide polymorphism that occurs in exon 1 of the gene. So that results in an adenine to guanine substitution, which ultimately results in asparagine to aspartic acid replacement. And I just want to note that there are a few different SNPs or single nucleotide polymorphisms that occur within the mu opioid receptor gene. But in terms of allelic frequency, this is the most represented. And it occurs in this putative glycosylation site, which is thought to influence the trafficking and signaling of the receptor. So as I mentioned, this is a fairly common SNP, though there is some variation depending on ancestry. We see it most highly in people of European and Asian ancestry, a little bit less in African American and Hispanic populations. There have been a few studies that have looked at kind of expression of the mu opioid receptor in individuals who have this mutation. And in postmortem studies, they've actually found there's reduced mRNA expression of the mu opioid receptor in people who have the G allele. And then functionally, there is some evidence that this SNP reduces response to opioid-induced analgesia and alters susceptibility to drug dependence. Though there is some, there's not a consensus on whether or not that altered susceptibility is actually a risk factor or a protective factor. And there are some sex-dependent associations that might be involved in there as well. But most importantly for the study that I'm going to describe to you today, there was a clinical study that found that infants who are exposed to opioids in utero and who are carrying that G allele have shorter hospital lengths of stay. So what that suggested to us is that perhaps this SNP might have some protective effects against either this prenatal or early life exposure to opioids. So luckily for me, our lab had already generated a transgenic mouse line that has the equivalent SNP in the mouse mu opioid receptor gene. So it's the same adenine to guanine substitution that's at a slightly different nucleotide residue, but it results in the same amino acid replacement. And so a postdoc, Steve McGew, who generated this mouse, also characterized the effect of this mutation on expression of the mu opioid receptor throughout these brain regions that, again, we know are highly associated with reward. So the ventral tegmental area, the nucleus accumbens, that's part of that mesocorticolimbic dopamine pathway. And then also with pain and withdrawal, so the periaqueductal gray and the hypothalamus. So across those regions, we're seeing reduced mRNA transcripts for the mu opioid receptor gene in animals who are homozygous for that mutation. That also translated to protein expression. So here, this is in the thalamus, we're seeing reduced protein levels of the mu opioid receptor. And then lastly, when looking at binding, here he used Damgo, which is a synthetic opioid that has very high specificity for the mu opioid receptor. We're seeing reduced binding in tissue from animals homozygous for that SNP. So clearly there seems to be some loss of function in terms of expression of the mu opioid receptor in these animals. Additionally, when we look at some of these behavioral assessments of responses to opioids, we're also seeing some changes in these mutant animals. So on the left is a locomotor sensitization assay. And on the first 3 days, all the animals receive saline, and then beginning on day 4, this is when they are either treated with morphine or the controls continue to get saline. And what we're looking for in this development of sensitization is an augmented locomotor response to the drug over time. And in our wild type animals in the triangles here, you see that after repeated administration of morphine, they do show that elevated locomotor response, suggesting some alterations, neuroplastic changes in response to that drug. Whereas our mutant animals, the GGs here in the circles, they do show an initial locomotor response to morphine, but it never sensitizes, suggesting that there is some disturbance in their ability to develop that sensitization. And here on the right, we're looking at antinociception in response to morphine, so a hot plate assay. We see that our GG animals, again, are showing a reduced kind of efficacy in response to morphine analgesia, right? So in both sensitization and antinociception, our GG animals seem to be responding differently, which does seem to fall in line with some of the human data with this SNP. So using this mouse line, I wanted to then shift into that early life paradigm to understand how might changes in the receptor be influencing that early life exposure and withdrawal to opioids. So there's a fairly large literature looking at early life exposure, and there does seem to be a consensus in some of these behavioral outcomes that are seen. So there's clear evidence that early life exposure delays emergence of postnatal reflexes in rodents, induces a somatic signs of withdrawal upon abstinence, and that can either be spontaneous withdrawal, so just not treating them with the drug anymore, or inducing withdrawal through the administration of an opioid receptor antagonist. And then lastly, there is some evidence that it can, this early life exposure to opioids can increase susceptibility to drugs of abuse later in life. So altered responses to drugs of abuse in either adolescence or adulthood. So what I wanted to see was whether or not using this transgenic mouse line, could we determine if the OPRM1 SNP is influencing some of these outcomes. So one of the challenges of modeling neonatal opioid withdrawal syndrome in rodents is that the developmental timeline of a mouse is obviously very different from a human, but notably the gestational period, when you look at kind of central nervous system development, a newborn mouse is most analogous to a third trimester human fetus. So what that means is if you only do gestational exposure in a mouse, you're kind of missing out on this last trimester of a human pregnancy. And if you only do the postnatal period, you're missing out on kind of the first and second trimester. For this study, we decided to use this postnatal exposure paradigm for a few reasons. One being that it removes the confound of kind of variable in utero exposure. So we're injecting each pup separately. We know they're getting the exact same amount of drug. There is also some evidence in the human literature that the third trimester of a human pregnancy is a sensitive period for the development of NOWs. So infants who are exposed in that last trimester are more at risk of severe neonatal opioid withdrawal syndrome symptoms compared to infants who are exposed in the first 2 trimesters. So we felt that it was a good starting point to evaluate some of these effects, both looking again at that acute withdrawal period and throughout the lifespan, well, the early lifespan of the animal. So our studies were looking at both kind of the developmental milestones within the first 2 weeks of life, in addition to that withdrawal period right after cessation of drug treatment. We also looked at some affective behaviors in kind of this early adolescent stage. So that's another period of kind of synaptic growth and pruning, and a sensitive period. We wanted to see if that is being affected by this early exposure and withdrawal. And then lastly, in adulthood, we assessed these animals for changes in drug sensitivity. So what was nice about our model was that we could look at that early withdrawal period, but also map out the effects on these later stages of life in these different kind of endophenotypes, affective behavior and drug sensitivity. So starting off, we looked at just weight gain in those initial few weeks of life. So again, they're being exposed from postnatal day 1 to postnatal day 14. And again, this is a period of rapid growth in all of the animals. However, we do see a reduction in weight gain in the animals who were treated with morphine. And that was seen in both our males and females of all 3 genotypes. When looking at development, we wanted to do a battery of kind of developmental milestones to assess how that morphine exposure was affecting their developmental trajectory. So we tested them in 3 different reflexes, I guess you can call them. So the forelimb grasp, this is assessing kind of strength and muscle development, so their ability to kind of hold on to this thin wire for at least a few seconds. Surface riding, so their ability to flip from their back onto their front within a second. And then this last one is this pivoting behavior. So in the first few weeks of life, both mouse and rodent pups kind of walk in a circle because their hind legs are not quite as developed as their forelimbs. So over time, they should be able to walk in a straight line and be able to go outside of this circle. So we were also assessing kind of that pivoting behavior. And again, similar to the weight data, we found that animals who are treated with morphine, regardless of sex or genotype, did show this delay in achieving those developmental milestones. So in this case, we were looking for days to criteria. So how many days did it take them to achieve that particular milestone? And you can see in our morphine treated animals, it's taking them a bit longer to reach those milestones. To get at more of the affective components of withdrawal, we were interested in looking at isolation-induced ultrasonic vocalizations. So infant pups will emit these ultrasonic vocalizations, so they're outside of the range of normal human hearing, but we can utilize technology to visualize those calls. There have been several studies that have tried to translate this ultrasonic vocalization to a human infant crying. There is a component of kind of distress there. So typically when the pup is isolated from the dam and its litter mates, it will vocalize, and that prompts caregiving behaviors from the dam. There's also previous studies that have shown that if you induce withdrawal in a neonate that has been exposed to opioids, you see heightened levels of vocalizing. So it can potentially be used as a proxy for maybe that hyperirritability or excessive crying that I mentioned is a symptom in NOWs. So we assessed this behavior on PND15, which was 24 hours into the spontaneous withdrawal. And we found some interesting results here. So the males who were treated with morphine showed significantly higher vocalizations, whereas the females didn't. So this was kind of our first evidence of a sex-specific effect in this model, in that perhaps the males were experiencing a more severe withdrawal. It is important to note that ultrasonic vocalizations do follow a kind of ontogenetic profile. So from birth to about 2 weeks of life, you do see changes in how much animals will vocalize in this paradigm. So one interpretation is that this is reflective of kind of emotional distress during withdrawal. Another interpretation can be that this is another sign of a neurodevelopmental delay, right? So our males are still kind of vocalizing at a rate that a younger animal would exhibit, right? But either way, we're starting to show signs or see some evidence that males might be more affected in this paradigm. So in adolescence, we wanted to look at a few of these affective behaviors. So mood and anxiety disorders are highly correlated with early life adversity. And we were interested to see whether that early life exposure to drugs could be influencing baseline affective processing. And so again, we're using a kind of a battery, a few different types of assessments of affective behavior here. One is the marble burying task. So rodents, their natural inclination is to kind of bury novel objects that they're not familiar with. But if you treat them with a benzodiazepine, which is an anxiolytic, or SSRIs, which are antidepressant drugs, you see reduced burying. So it's been interpreted as a test of anxiety-like behavior. Similarly, the elevated zero maze is based off a rodent's natural aversion to open spaces. So typically you would see an animal spending most of its time in these enclosed arms of the EZM. However, again, with treatment with an anxiolytic or an antidepressant, we'll see animals spending more time on these open edges. So when looking at our animals, we again saw this interesting effect in the marble burying task. So only in our wild type animals, morphine exposure significantly reduced the number of marbles that were buried, both in the males and the females. And so again, one interpretation of this is that our morphine treated animals, specifically in the wild type, are showing reduced anxiety after exposure to morphine. However, when we looked at the EZM, we didn't see a similar effect. So it's possible that we're not seeing a generalized change in all anxiety, but perhaps a change in response to environmental cues or to novelty. So there have been some studies in looking at other neurodevelopmental or animal models of neurodevelopmental diseases, including models of autism spectrum disorders that have used marble burying and seen a reduction in that marble burying, and they've referred to it as kind of changes in environmental processing. So we are still trying to understand exactly what this might mean here. But again, we're seeing a morphine effect that is only in our wild types and seems to be protected against in our animals who have that G allele. And then lastly, in adulthood, we again wanted to assess how these animals are responding to opioids. as an adult if they had that exposure in the neonatal period. The locomotor sensitization assay is associated with changes in dopaminergic neurotransmission, and again, like neuroplastic changes that are occurring in that mesocorticolimbic dopamine reward pathway. It doesn't necessarily map directly on to more operant behaviors like self-administration, but it can give you a sense of alterations in how the animal is responding to the drug. So here we're looking again at our males in the 3 different genotypes. The open circles are the animals who were exposed to saline as neonates. The dark circles are the animals who were exposed to morphine as neonates. The baseline just represents a period where everybody got saline in the assay, just to establish kind of baseline locomotor activity. And then morphine 1 is the first day of that exposure to morphine, and then morphine 4 is that 4th administration. So really trying to assess that development of sensitization. And what you'll see here is that although both the saline and the morphine pre-exposed animals developed sensitization, we see this exacerbated or augmented effect in the animals who were pre-exposed to morphine, suggesting that they are having this altered sensitivity to morphine as an adult. Interestingly, we did not see that effect in our heterozygotes. So they developed sensitization, but there was no difference between the animals who were pre-exposed to morphine. And then in our homozygous GG animals, this was actually replicating the original data from the Magoo paper, where the GG animals just don't develop sensitization at all. So just focusing, I guess, on the wild types versus the single allele, it's possible that having the G allele is protecting against that heightened sensitivity. Interestingly, we saw exactly the opposite effect in our females. So females who were exposed to morphine in the neonatal period actually showed a reduction in sensitization compared to the animals who were exposed to saline. But again, this effect is only in the wild types. We don't see it in our heterozygotes or the homozygote mutants. So I mentioned that the locomotor sensitization, it's kind of your first pass for trying to understand changes in drug sensitivity, but not necessarily getting at the rewarding or motivational aspects of drug seeking. So in that 2009 paper, we had shown, or it had been shown that even though the GG animals don't express sensitization to morphine, they will still show a place preference for morphine. And interestingly, there was a sex by genotype interaction here where the female GGs were the only ones who did not exhibit a place preference. And then on the right, this is data from another group who used the same transgenic line of animals in a heroin self-administration paradigm and found that animals with the GG allele would nose poke more for heroin compared to the wild type animals. So future directions for this project, we are really interested in understanding, whether or not this alteration in sensitization is mapping on to differences in kind of the rewarding aspects of the drug. But in summary, we found here that neonatal morphine exposure delays emergence of developmental milestones, regardless of the sex or the genetic variant. This is really interesting, again, because we were using this postnatal paradigm. I think it really lends credence to using that model to really dive deeper into some of these mechanisms underlying the development components of the morphine exposure. We saw that it increased ultrasonic vocalizations only in males during spontaneous withdrawal, which again might speak to the developmental delay or an increased severity of that withdrawal syndrome in the males. The OPRM1 SNP protected against the effect of neonatal morphine exposure on behavior in the marble burying task, which could potentially suggest some influence of the SNP in kind of environmental or kind of response to environmental stimuli. And then lastly, we found that the neonatal morphine exposure alters the development of adult morphine locomotor sensitization in a sex and genotype dependent manner. So altogether, you know, what we found here was a little bit in contrast to our original hypothesis, which was that the OPRM1 SNP would reduce the severity of the withdrawal. It seems like it didn't really impact the developmental effects per se, but the long-term effects is really where we started to see an interaction between the genotype and the opioid exposure, which I think really highlights the importance of studying both the early withdrawal, but also kind of these long-term outcomes. And then these sex differences, I think, are really interesting in the context of a recent paper by Borelli et al. And Cameron Bryant's group, showing that there are sex differences in kind of transcriptomic neuroadaptations that are happening in a very similar postnatal opioid exposure paradigm. And so it's possible that this opioid exposure, morphine exposure, is inducing some genetic changes that might contribute to long-term outcomes. So I just quickly wanted to talk about some things that my lab is now expanding on. So we are really trying to further characterize this spontaneous neonatal opioid withdrawal phenotype, both in wild type animals, but also can be easily applied to these other transgenic models. So one of the limitations of our study is that we only looked at one time point, so 24 hours into withdrawal. But this is some preliminary data looking at multiple time points. And we actually found that when we look at ultrasonic vocalizations, those seem to peak at 8 hours post-withdrawal as opposed to 24 hours. And that kind of brings into question, you know, are we really capturing a period of time where the animals are showing the most severe withdrawal syndrome if we're only looking at 24 hours? And in addition to that, we're trying to implement some deep machine learning to more closely analyze the vocal or the ultrasonic repertoire that's occurring at these different stages or different time points of withdrawal, to see if that can give us a little bit more understanding of that, the emotional processing that's happening. And we also want to incorporate some other kind of measurements of withdrawal that can be happening during this period. So here we're looking at thermal sensitivity, which could be an indicator of hyperalgesia in these animals. So there was actually a recent clinical paper that showed that in humans, infants who were exposed to opioids in utero show this heightened pain response kind of within that 24 to 72 hour period. So here we're also showing that our morphine treated animals have this lower latency to respond to a thermal stimulus, and that is extending for quite a period of time, up to 48 hours after the last injection. We are also utilizing a precipitated paradigm as well. So trying to map out behaviors in our neonatal mice that we can use to, again, more clearly define what that physical withdrawal syndrome looks like. So here's an example of wall climbing and jumping that we see in our neonatal animals, and some other behaviors that have been shown to be distinct to withdrawal in young animals as opposed to adult animals. So again, really trying to define that. And in that precipitated withdrawal phenotype, again, we see when we look at our OPRM1 SNP animals, again, this is just preliminary, some preliminary data, but it seems like the precipitated withdrawal is similar among our different genotypes, in addition to this hyperalgesic phenotype. But what was really interesting was that this hyperalgesia, so the latency to respond to the hot plate, correlates pretty well with the withdrawal score or the withdrawal severity. So it's possible that kind of just pain response during this withdrawal period is a good predictor of the physical withdrawal. So that's something that we're really interested in pursuing in my lab as well. So trying to understand some of the nociceptive changes that are happening in these animals. And so with that, I just want to acknowledge everybody who contributed to this project. So Julie Blundy, who was a fantastic mentor during my postdoc, Michelle Ehrlich, who collaborated and provided some really helpful insight. And then I just also wanted to quickly acknowledge my many students in my lab who I didn't have time to talk in detail about their projects, but are also helping to kind of expand on those neonatal opioid withdrawal studies. And so with that, I'm happy to take any questions.
Thank you, Dr. Robinson. I guess we'll start off by one, listening to all your virtual applause that's happening around the country, and two, by opening up the floor for any questions. Feel free to unmute yourself if you have a question for Dr. Robinson.
Hi, Dr. Robinson. Very nice talk. I really enjoyed it. You showed a piece of data toward the end indicating that it looks like, depending on looking at 8, 24 for 48 hours, it seemed like the increase in USVs was disappearing. Mm-hmm. And so I know you mentioned earlier that it's kind of a debate whether the USV phenotype is withdrawal-based or neurodevelopmentally based. That piece of data to me suggests that it might actually be a withdrawal phenotype. I'm wondering if you had any thoughts on that. Yeah. And then I have one more question after that, if you don't mind.
Yeah, so I agree. I think mapping out the kind of temporal expression of the ultrasonic vocalizations does suggest that it might be related to that withdrawal experience. I think particularly when you combine it with these other measures, that's when the data becomes more convincing, right? So that we're still seeing this hyperalgesic effect, and then adding on kind of these somatic withdrawal symptoms. I think it's something that could be contributing to the withdrawal syndrome. And then I don't have the data for it here, but the fact that precipitating withdrawal also increases the ultrasonic vocalizations suggests that it is, again, representative of kind of the aversive components of the withdrawal. But I think having just that data by itself, it's hard to separate it from some of the developmental components as well.
And then one more, thank you. And then one more quick question. As you know, we're using a very similar protocol as what you described. And we're seeing very little evidence actually for long-term effects. I can't remember if we've looked at opioid exposure in adults, but we've looked at several phenotypes and they're very subtle, if not, even significant. But I'm wondering, we see a recent piece of data that we've collected, we see very big differences in myelination occurring during the withdrawal state at P15. And I'm wondering if you're considering looking at perhaps older mice, like aged mice, and the much longer term effects that might occur after this exposure period.
Yeah, no, that's a great point. I haven't, in my work, I haven't looked at kind of that neonatal exposure in aged mice. We've kind of been focusing on that adolescent and adult period. And we have seen, this locomotor sensitization effect. We've looked at some cognition. Novel object recognition or tactile recognition, and have seen some long-term effects as well. But I agree, it might be a case here where it's very specific behaviors that are being affected, and it's not a kind of just a broad changes in behavior. The other thing that we've considered is that it might not necessarily be strong changes to baseline behavior, but perhaps exacerbating responses to stressful stimuli. So that's something we're interested in exploring too. So if these animals are exposed to another insult, do you see kind of an exacerbated response? But I need to think more about the aged animals, because I have some mice that have just been sitting here for a while, so that might be worth checking out.
Great, thank you. Nice talk.
Dr. Crab.
Hi, Shivon. Really interesting data, and the fact that you started to look over the full first 48 hours, at least, of the of withdrawal syndrome in the infant adolescent mice leads me to wonder if you've thought about what would happen if you, it might be clinically relevant to suppress that withdrawal one way or another. And then, which you could also do in control animals, with a long-acting opioid, or even with an antagonist, and then see if that affects the some of the differences that you do see in adulthood.
Yeah.
Desensitization.
Yeah, so that's definitely a huge interest, and a project that some of my thesis students are working on now is, can we now use this model to assay different therapeutic interventions? And if we can reduce the withdrawal, does that also prevent some of these long-term effects that we've identified from happening? Because I do think that's an important translational or clinical question as well. Is it enough to just treat the withdrawal syndrome, or is it the fact that they've been exposed during this very sensitive developmental period? Are there other interventions that we need to think about?
Thanks, Dr. Robinson. Simona, you have your hand up?
Yeah, thank you so much. This is such a cool talk, and so many really cool findings and a really great discussions. There's just so many more questions that I want to ask based on the discussion. Oh, I should say I'm a graduate student in the Doherty lab. So a bit younger than most of the people who are asking questions, but this really sort of made me think about, so we actually just published a study where we did prenatal exposure only to oxycodone, and then pre and postnatal exposure to oxycodone. And we actually found that the postnatal exposure had effect above and beyond the prenatal exposure, which really matches with your data really well. And now I'm thinking, wow, we should have just done the postnatal too, but that would have too many groups, probably too big an insight. But sorry. So the question I wanted to ask is, you've obviously collected a lot of really cool in-depth USV data, and I was wondering if you're thinking to do a more in-depth analysis of those USVs, not just looking at the total number, but maybe the pitch or the range of the USV, how loud they are. Because I know we've noticed some really interesting findings when you start to parse out the loudness, for example, is something that we're thinking to compare to the loudness of the calls of these babies in the NICU, for example.
Yeah, I think that's a great idea, and something that we're definitely interested in. So I have some students who have been really excited to work with machine learning algorithms, so programs like Deep Squeak, for example, And they've been doing great work in kind of optimizing those, like the training process and getting it to the point where we can just feed in that USV data and get things like that. So like, you know, how loud it is, the duration of the calls, but also kind of like the repertoire. So the combination of the different, like the qualitative, I guess, component of the calls, and is that changing during the withdrawal period? That could potentially be a way to differentiate like what is a normal isolation call versus a withdrawal call, right? Like, are those qualitatively different? And then to your other point about kind of the prenatal exposure plus the postnatal exposure, I had done some pilot studies with that as well and saw something similar, that the postnatal, just the postnatal alone, in terms of the kind of like those developmental milestones and the weight. I didn't do the withdrawal studies as in-depth as this work, but it did seem very similar to just the animals who were treated postnatally alone. So I do think that speaks to, again, you know, like these sensitive periods, and that getting the morphine exposure during the postnatal, those first few weeks of life might be a way to model kind of like a more severe syndrome.
Thanks, Dr. Robinson. I'm really glad that you all are looking at USVs. I think it's one of the most unique behavioral measures that we have, and I think it's important to characterize, you know, them fully. So that's great. I do have a question in the chat. So I'm asking this one on behalf of Elizabeth Yen. She's asking Dr. Robinson, well, she's complimenting you. Dr. Robinson, thanks for a fascinating talk. Are you planning on following the somatic growth beyond P21? Did you notice any difference in their feeding behavior during withdrawal, early versus the end of withdrawal in particular?
Yeah, that's a great question. So I have some data from students in my lab here who have shown that the weight changes, so that reduction in weight gain persists into adolescence. So at least that component seems to continue past P21. The feeding behavior, I think, is a really interesting question, and something that I've thought about in, you know, are some of the effects that we're seeing just due to the pups being smaller or malnourished? And, you know, again, thinking of therapeutic interventions, could supplementing the morphine treated pups with, you know, like a high fat diet or something to kind of bring their weight back up, could we maybe mitigate some of the neurodevelopmental effects? But I don't have that data. I think it's a really interesting question, and also a little bit of a tangent, but could possibly tie into the effects of opioids on the microbiome, some of the gastrointestinal effects that we see in the clinical populations. So I think there's a lot of interesting work to be done with feeding behavior and kind of those more physiological measurements.
That was great. Definitely a strong impact of opioids on the GI system that might interact with these behavioral phenotypes in interesting ways, for sure. That's cool. Are there other questions? I have a question, which is probably just based on my own ignorance. But it has to do with the A112G mutation. You mentioned just briefly that is putatively located in a glycosylation site. And so I had two questions related to that. One is, how do you get from a mutation in a glycosylation site to a reduction in transcription and expression of that gene? And then I guess it's not really related, but a follow-up to that is, does that decreased, it seems like that decreased expression would just reduce sensitivity to opioids in general. But your data doesn't suggest that. You seem to have, like you mentioned, you showed us this time dependent effect where the not as many differences during the neonatal period, but you do see what looks like a decrease in opioid sensitivity in adults.
Mhm.
So what's up with that? Is there a dynamic change in how these expression differences are manifested, or is there something else going on in terms of signaling? I'm just wondering if you can comment on the complexity of your results.
I can definitely comment. I can't say anything definitively. But so to your first question, how does this SNP in a glycosylation site affect expression levels? So there is some evidence that, particularly in GPCRs, changes in these glycosylation sites can affect the trafficking of the receptor to the membrane. So it can change the cell surface expression of the receptor. I don't know if this is directly linked to the glycosylation site or issue, but there has been some hypothesizing that the SNP might result in an mRNA transcript that is not fully effective, I guess, in being translated into a protein. So that could contribute to the reduced protein expression. So yeah, I think there's still a lot of questions on how exactly that mutation is causing this loss of expression. But it seems, again, to map on really well to at least the data that we have from humans, where in postmortem tissue, we see less expression. And there are also some neuroimaging studies, some PET scans that have shown reduced binding to mu-opioid receptors in people with the GG allele. So yeah, I guess more work needs to be done to understand the cellular mechanisms that are causing that SNP to reduce the expression. And then the question of, it seems like it would reduce sensitivity to opioids. I think it does. So the initial characterizing data, we see less of that anti-nociceptive effect in the hot plate and that lack of sensitization. But it could be that the reduced sensitivity might lead to increased drug-seeking behavior, like the animals want or need more of it to get the high. So that's why we see this increased self-administration. And whether or not that neonatal morphine exposure is having, epigenetic changes on the activity of the receptor is unclear. I think that's some additional work that could be done using this model, since the initial characterizations were just looking at adults who had never had any previous exposure. So yeah, I think there's a lot of different avenues to go in to, really understand how this genetic variation is influencing these outcomes. And the one other thing I'm just going to throw on there, you didn't ask, but something I think to keep in mind is that initial study I mentioned that said human infants who were exposed to opioids and had that G allele had shorter lengths of stays in the hospital. We initially interpreted that data as, okay, the SNP is protective. It might just be that the SNP increases the effectiveness of the pharmacological treatments, right? They respond better to the first line therapies with that SNP. So I'm very interested to see, whether if you use some therapy or pharmacological intervention in this model, would we see more reduction in withdrawal in our GG animals, even though at baseline they don't show any differences?
That's great, obvious impacts for precision medicine. Cameron Dr. Bryant has another question that he put in the chat, so I'll ask on his behalf.
I can read it. It's fine.
Oh, I was going to do
curious.
my Cameron impersonation.
Go ahead. I'm just curious if you've had a chance to look at differences in maternal care and the morphine versus saline injected pups. Like, our concern, because again, we're using the same model basically, is that maybe that they're just not tending to the morphine pups as well.
Yeah, I think that's a great concern. I have not empirically measured that. The only time I noticed what seemed to be like a difference in the maternal behavior was when we had, because I had originally done a pilot experiment where it was a split litter design. So half the animals got morphine within the litter, and those animals were just, the morphine treated animals were just much more likely to die in the split litter design versus if it was just a single saline or morphine. And I think it was because the mom was like preferentially, using her resources for the pups that looked healthy versus the ones that didn't. But that is something that I'm interested in exploring too. If in this particular model, if there are differences in maternal care, is that something that you could manipulate, right? And again, reduce some of these phenotypes.
That's interesting. That could very well explain why we had so much death in our, because we used a split litter design. So.
Okay, we're actually at exactly an hour, and it'd be fun if we actually finished in an hour for once. I want to end just by saying thank you for coming to give the talk. It was a great talk. You know, you go to talk sometimes where you just feel like you're in good hands. The speaker is taking care of you and presenting the complex information in a very clear way. You did that was just an extremely nice talk. And one of the best question and answer sessions that we've had. So I really appreciate it. It was an absolute joy having you speak to us today.
Thank you so much. Yeah, that was a great discussion. And I'm happy to follow up if anyone has additional questions or want to chat more about this model.
Great. And thanks everybody for coming. Everybody have a great rest of your day.
Thanks,
See you next month. Hi, John. Good to see you.
Good to see you.
Thanks again, Shivon. That was great.
Oh, it was my pleasure.
I'll thank you again via email, but
Okay.
that was awesome.
Sounds good. Have a good one.
Bye, Shivon.